Well, it's 12:30. A lot of people still have yet to join, but I want to get started because we have a lot to do today. Not a lot of things, but a lot of the same thing. Starting a new topic. So let me share my screen with you. You see the overview of where we're going?
Yes, we see it.
Okay.
Yes.
All right, this is what we're going to be doing for the rest of the semester. These 4 topics, plus maybe one more on climate change, but that will be optional. So we're going to be talking about energy, enzymes, photosynthesis, and respiration. This is what life is all about, these 4 topics, and everything we've done up to this point has been to provide you with background so you can understand how these complex systems work. So we're going to talk about energy, what it is, where do we get it, what are its forms, enzymes, how do they work, why are they essential for life, photosynthesis, which is where all of our food comes from, and respiration, how we get energy from the food we eat. Well, energy. Life exists on this planet because the sun shines. We get all of our energy from the sun. And the sun's energy is captured by plants and it's consumed by animals. And between the two, nutrients are cycled. So photosynthesis provides energy in from the sun, and respiration provides energy out from the products of photosynthesis. So what is energy? It's the capacity to do work. What is work? The ability to move matter in a direction it would not move if left alone. So, energy is measured in calories. The definition of a calorie is the amount of energy necessary to raise the temperature of 1 gram of water 1 degree centigrade at sea level. Now, you're all familiar with calories. Anytime you talk about diet, you're talking about calories. But those aren't really calories. Those are kilocalories. A kilocalorie is 1,000 calories. So if you're talking about a diet with 2,000 calories per day, that's really 2,000 kilocalories, which translates to 2 million calories in a normal diet. Well, the forms of energy, potential and kinetic. Potential is stored energy, like a rock sitting at the top of a hill, or ions on a side of a membrane. And we'll come to this term chemiosmosis later. It's a very important term. Kinetic energy is the energy of motion. So when the rock gets pushed from the top of the hill and tumbles down, that is kinetic energy. Or ions, instead of sitting on one side of a membrane, if they move through it, that is kinetic energy. And we'll talk about that with regard to chemiosmosis. Potential energy and kinetic energy are being constantly interconverted in living systems. To understand this, we need to take a quick peek at the laws of thermodynamics. The first law is that in a closed system, energy can be neither created nor destroyed, but it can change in form. And to put that on macro scale, the amount of energy present at the beginning of the universe will be the amount of energy at the end of the universe. The form may be different. And there are a number of hypotheses about how the universe is going to end. I don't want to take time to go through them now. None of us will be here when that happens, and nobody really knows how it will happen. If you have any questions or comments as I go along, please stop me, or if I go too fast, let me know.
Okay, so Professor, you said that the rock at the top of the hill has energy. I mean, I understand how it has it when it falls down, but how does it have energy just sitting there?
Its energy is called potential. So the potential, the energy itself lies in the mass of the rock. So between the mass of the rock and the force of gravity, there is potential energy. When gravity pulls the rock down the hill, that potential energy is converted into kinetic energy.
So you're saying it's like energy because it's sitting there, it's to hold it there?
Right, it's got energy, but it's only potential. It's not being expressed.
Okay.
And I'll illustrate that more clearly in a little bit.
Okay.
Okay, so the second law says that energy transformation results in increased disorder. And some of the energy is lost. It's no longer available to do work. That brings up the term entropy. Entropy is a measure of the amount of disorder or randomness in a system. And all living things are systems. So the implication of the second law is profound. It says that disorder is always increasing, or chemical reactions go to equilibrium. And the only way disorder cannot continue to increase is if there's an energy input into the system. But when are biological systems at equilibrium? When they're dead. Life requires a constant input of energy, and if that energy input stops, the organism dies. Why do cells need energy to survive? Well, I've just told you that because of the second law of thermodynamics. So cells are subject to becoming disordered. They remain alive and healthy only if they have a constant input of energy, and that requires chemical reactions in a cell. Before going to that, I'd like to take a peek at electromagnetic radiation. It is a form of kinetic energy, and it consists of a spectrum of waves. Now, this is always a hard one for me to comprehend, that a wave also has the properties of a particle. And that particle is called a photon. And when we come to discussing photosynthesis, we will talk about photons. Each photon carries energy. Now, this is an illustration of the electromagnetic spectrum, which goes from low energy on the right to high energy on the left. And that corresponds to long wavelengths on the right and short ones on the left. So these are the long wave radio waves, microwaves, infrared waves, ultraviolet, X-rays, and gamma rays are the shortest in length and the highest in energy. Now you see this little clear box in the middle here? That represents visible light. Visible light means the light that we can, the wavelengths that we can see with our eyes. And they vary by color. The longest ones that we can see are red. Shorter ones are yellow, then green. And the longest wavelengths that we can see are blue. Interestingly, plants can see the same spectrum of lights with one exception. Well, why are plants green? We go back to the previous slide. We have the four major colors: red, yellow, green, and blue. If we look at what makes plants green, it's chlorophyll. And where does chlorophyll absorb? You see the wavelength spectrum here at the bottom. Chlorophyll absorbs in the blue region. If you get longer wavelengths over to the green region, it does not absorb. It reflects. And then if you get down into the longer waves, the red region, it again absorbs. So plants can see red light and blue light. They cannot see green light, so they reflect it. And so we see plants as green. Now, not all plants are green. So I want to make a significant point about this, because it has to do with what you see every day when you go out. You see that most plants are green, but you also notice now and then a plant that isn't green. What are the reasons a plant might not be green? Well, one is that it really is green. It has chlorophyll, but that pigment is masked by another pigment. You've all seen, for example, these ornamental red maples. They're green trees, but the green is masked by a pigment called anthocyanin. Another possibility for why a plant isn't green is that it's deficient in something like nutrients. And so its leaves turn yellow. That doesn't mean it doesn't have chlorophyll, but it has other compounds that are blocking the chlorophyll. And if they're chlorotic, usually there is much less chlorophyll. There are mutations which can cause albinism, inability of a plant to make chlorophyll. Here's an example of an albino corn seedling, pure white, and it will die because it has no chlorophyll. You can keep it alive by feeding it sugar, but eventually it cannot make its own food. Another reason a plant might not be green is that it's deciduous. and the season is the fall, and chlorophyll is degraded, replaced with other pigments which fall off to be regenerated again in the spring. So these are a number of reasons why a plant that you see might not be green. But another very interesting reason is that the plant you're looking at is parasitic. Some plants make no chlorophyll at all. Instead, they make their living by parasitizing green plants, which do have chlorophyll. And here's a picture from East San Diego in the desert. Here's a green tree, and here is a yellow plant growing on it. The name of this plant is dodder. Here you see an up close of it, and it is growing on the green plant. It's a parasite. It will eventually kill that plant. The way it gets food from the plant is by these little tendrils which adhere to the green plant. They make little bodies that penetrate the surface of the plant so that they can absorb nutrients from the plant. So they do nothing for the plant. They derive their living entirely from the plant, and the plant suffers. So parasitic plants are another reason why you might see plants that aren't green. And again, just to remind you, here are several of the reasons that plants might be green. And in addition to that, it might be parasitic. And these are all things, except maybe the albino, all things that you will see when you go out and about. Okay, the function of the non-visible radiation. I won't go through these in detail, but gamma rays are very energetic. They're used for a number of things, including cancer therapies and tracers. X-rays, you all know about. They pass through living tissue, leaving shadows where there are bones and organs. Ultraviolet radiation tans your skin, interacts with vitamin D. It's often used to sterilize entire rooms, for example, to get rid of coronavirus. Then you have the visible, which we've talked about. Infrared is shorter. You'll find it are used for wireless communication, like garage door openers and TV remotes. Microwaves, you probably use them every day. They heat food by vibrating fat molecules and water molecules. And finally, radio waves are truly amazing. They're the longest in the electromagnetic spectrum, can range in length from a millimeter, which is very small, to 100 kilometers. 100 kilometers is 60 miles. That is a long way. One wave in a radio wave can be many miles long. And radio waves are used for the long-range transmission of radio and TV signals. Okay, that's an aside just for your information, how electromagnetic radiation affects you in your daily life. Okay, so then how is the energy in an electromagnetic spectrum captured and used so that life can exist? We're going to spend a lot of time on that. The answer is photosynthesis. And we'll get to photosynthesis eventually, but first, what is it? It's a process performed by plants, only plants. It's capable of capturing the energy in a part of the electromagnetic spectrum that we humans see as visible light. As I said, plants see the same wavelengths that we see. Now we're going to leave photosynthesis aside and come back to it later and talk about chemical reactions and metabolism. What is metabolism? It's the sum total of all the reactions in a cell. So in a living cell or a living organism like you or me, at any point in time, thousands of chemical reactions are going on. And every reaction in a biological system is mediated by an enzyme. There are no biological reactions that are not catalyzed by an enzyme. They speed up the reactions of a cell. Without them, chemical reactions on their own would be way too slow to keep a cell alive. So how does a series of chemical reactions work? You have substrates A and B. These could be any molecules. And you have an enzyme, which converts them to compound C. Then another enzyme converts C to D plus E. Then a third enzyme converts D plus E to F. So this is a metabolic pathway catalyzed by enzymes. So the essence of life, again, we're coming back to a brief mention of photosynthesis and respiration, but this is how energy works in living systems. So there are two types of chemical reactions, endergonic and exergonic. Endergonic require energy input. Exergonic release energy. So you see that in photosynthesis and respiration. In photosynthesis, energy comes in. Where does it come from? It comes from the sun. So plants can take energy from the sun and can combine it with carbon dioxide and water and make sugar plus oxygen. When we eat food, the opposite occurs. The food we eat is broken down till it finally becomes something comparable to glucose, which is then undergoes reactions to release carbon dioxide and water plus energy. So life is all about energy in from plants, energy out when animals eat food. Now you asked about kinetic potential and kinetic energy. We can look at these in terms of without an enzyme present or with. So in the top figure, this is a reaction without an enzyme. So you have a substrate, sucrose, which is a disaccharide, and it wants to be broken down into monosaccharides, glucose and fructose. Well, without an enzyme, it takes a lot of energy to do this, and that's called activation energy. So activation energy without an enzyme is high for this reaction. And then there is net energy released when you split this molecule into two. Now, if you add an enzyme, sucrase, specifically converts sucrose to glucose and fructose, now the activation energy is much lower. Now notice that the net energy is the same. Net energy is this amount in both cases, but the activation energy with an enzyme is much less. So what do enzymes do? We're going to talk a lot about that now. they orient the substrate and induce strain in the substrate. And I'll tell you how that happens. This blue blob is an enzyme. The smaller objects are potential substrates. And in this case, only the yellow one and the red one are really substrates. The green one isn't. So the yellow and red can fit into this enzyme and make a complex. The enzyme acts and out comes a product. The enzyme itself is not changed. I've shown you here the tertiary structure of an enzyme. It changes shape and then becomes the same tertiary structure again after the reaction is over. Enzymes and substrates are sometimes thought in terms of locks and keys. Shape means everything in enzyme reactions because enzymes are shape specific. Each enzyme can react with only one kind of substrate. And that's why in our cells, we have thousands of different enzymes catalyzing thousands of different reactions. So an enzyme's shape determines which substrate will fit. And this is often made analogous to a lock and a key. We imagine a lock being the enzyme and the key being the substrate. Then if the key fits the lock, it'll fit into the active site of the enzyme and a reaction occurs. But here on the right, if the shape is correct, it fits. If it's an incorrect shape, you see at the bottom, it doesn't fit and there's no reaction. Now, how do enzymes lower the activation energy? They do it by breaking bonds or weakening bonds in the substrate. So that's something to remember. And that's illustrated here twice in two different diagrams. The enzyme in the top one is yellow. The substrate is green. And there is an active site on the surface of the enzyme. So in this case, for example, this substrate might be sucrose. It's going to get broken into its two products, fructose and glucose. So when it binds to the enzyme changes shape. And I'll show you this more clearly later. Enzyme changes shape, and when it does that, it weakens the bonds in the substrate, in the sucrose, so that when the enzyme releases, there are two products instead of one substrate. And that's shown in a different way here at the bottom. The blue blob is an enzyme, purple is the substrate. When it enters, the enzyme changes shape and breaks bonds in the substrate. So the steps, first step, the substrate binds to an enzyme, that's step one here. The enzyme changes shape, you can see that best here in the lower diagram. The bonds in the substrate are broken or weakened, and then the products are released with little expenditure of energy. And I want to emphasize, or I want to remind you here about what are the bonds in a protein structure. In the case that the substrate might be a protein. They have hydrogen, ionic, hydrophobic, disulfide, and covalent bonds. And I'll come back to that in a little bit. So the mechanism of enzyme action, I'm going to show you the process several times because it's not an easy thing to grasp. So again, let's go through this in steps. The first step is that substrates here, red and green, enter the active site of an enzyme. Here's the active site. Here's the enzyme by itself, and here's the active site. So these substrates have the right shapes to enter into this active site. Form a complex. And the substrates are held in this site with weak interactions like hydrogen bonds and ionic bonds. Then the enzyme changes shape. And when it does so, the substrates are converted into products. Once that happens, the enzyme releases them as free products, and it goes back, assumes its original shape. It's ready now to do another reaction. This is called the induced fit mechanism. It's a model for how enzymes work. So this is just another illustration of what I've been telling you in the last 2 slides. Free enzyme and a free substrate get together, form a complex. The act of these of the substrate binding with the enzyme causes the enzyme to change shape, and when it does, it converts the substrate to, in this case, to products and releases them. This is known as the induced fit mechanism. The enzyme is induced by the fit of the substrates. Substrate rate effects. Enzyme reaction is a function of the substrate concentration up to a point. There is a maximum rate for any reaction. Without an enzyme, it's very slow. With an enzyme, it's very fast. And the maximum rate is reached at a fairly low concentration of substrate. Now, something very important for you to remember and understand how it works is enzyme denaturation. Enzymes can be damaged permanently or temporarily by exposing them to extremes of 3 things: temperature, pH, or salt. And this damage is called denaturation. Denaturation means that the tertiary structure of the enzyme is destroyed so that it will no longer function. So you see in the figure here on the left of the figure is a normal protein. Say this is an enzyme, and its tertiary structure with all the folds and the various features of a tertiary structure, and it's active. It can convert substrate to product. However, in the event that it encounters extreme pH or extreme temperature or extreme salt concentration, it will denature. And what that means is that the bonds that hold this structure together are broken, and you end up with only the covalent bonds. Covalent bonds hold the primary structure together. So what is this chain? It's a chain of amino acids, but it's completely lost its 3-dimensional structure and is therefore not active. Again, what are the bonds that break with these extreme environmental conditions? Hydrogen bonds are broken first, then ionic, hydrophobic, disulfide, and the covalent bonds are not broken. you'd have to have even more extreme conditions to break the covalent bonds. Now, in most cases, this is not reversible. Once a protein is denatured, that's it for that protein. In some cases, they can be renatured by giving them back a less extreme environment. Any questions about how an enzyme is denatured? What happens when denaturation occurs?
You said is it caused because of pH, temperature, and strength and solubility?
Ionic strength. So if you have a very high salt concentration bathing the enzyme, that will cause bonds to break and it will denature.
Okay, thank you.
Yeah.
So,
Go ahead.
So this is a Steve Stinson. So on a large scale, take for instance, if you pour acid on flesh or something and it starts to break down, that's actually what is happening. Denaturation is happening.
Yeah, if you poured acid on your skin, for example, it burns it. And the reason is that you're breaking bonds with a low pH.
That makes sense.
Okay, and I can make this a little more clear with examples. But what are the things that change the rate of a reaction? The temperature, the pH, and the salt. So the same things that will denature an enzyme can change the rate of a reaction. So on the left, we have two sets of graphs. At the top, we're measuring the rate of reaction plotted against increasing temperature from 0 C to 100 C. Now, a human body is 37 degrees centigrade. The optimal temperature for the typical human enzyme is 37 degrees centigrade. So the enzymes in our bodies are fine-tuned to function best at the temperature of our bodies. Now, if we go to other environments, for example, a heat-tolerant bacterium, the optimal temperature could be 75. And I've given you an example of that earlier with Taq polymerase, the DNA polymerase that's used in PCR. It will tolerate temperatures up to 95 degrees without denaturing. So whereas most enzymes would completely denature at 95 degrees, Taq polymerase does not. And why? Because this bacterium that produces the enzyme lives in a hot spring. The other example here is varying pH. Again, the rate of reaction plotted against varying pH from 0 to 10. This is the range of pHs that are possible. Now, in our stomachs, we find a pH of 2. That's very low. And there are a number of reasons why the pH of the stomach is almost like hydrochloric acid. And there are enzymes in the stomach that actually function. One example is a peptidase called pepsin that hydrolyzes proteins. And it has a pH optimum of 2. So it's perfectly adapted to function in the stomach. And if you put it anywhere else in the body, it either wouldn't function or it might completely denature. Now, on the other hand, sitting right next to the stomach is the small intestine, the duodenum, right beside the pancreas. The pH here is almost neutral, 6 to 7. There are other enzymes like the protease trypsin, which have a pH optimum that fits exactly with the pH of the intestine, approximately between 6 and 8. So all of the other enzymes in the digestive system have a pH optimum of the rest of the body. So is this clear? Getting back to someone asked a question about this. Temperature and pH determine the rate of reactions and determine when enzymes are active and when they denature.
Yeah, that's more clear. Thank you, Professor.
Okay. Anything else? All right, and I will go on. Most enzymes do not act alone. They have partners. These partners are categorized as cofactors, coenzymes, and prosthetic groups. What is a cofactor? Cofactors are inorganic, usually a metal. So iron, for example, copper and zinc, they're all cofactors. They're essential. We have to have these things in our diets, but we need very little of them. They're enzyme partners. Coenzymes are usually vitamins. Again, we need vitamins to be healthy. We don't need very much of them because they're recycled along with enzymes. And finally, what are prosthetic groups? They're non-protein parts of enzymes. So for example, there's a molecule known as heme, which along with hemoglobin binds oxygen and carries it in our blood. There's flavin, there's retinol. Flavin binds electrons. We'll talk about flavin later. So any questions about enzyme partners? They're non-protein molecules that enzymes require in order to function.
Now, I'm not sure, Professor, you said our body needs vitamins, but then you said it recycles them. So if that's the case, why do we have to take supplements?
Well, you shouldn't have to. If you had an adequate diet, you would replenish your vitamins every day. Different vitamins, some are water soluble, some are fat soluble. And so the water soluble ones are lost much faster than the fat soluble ones. So you're asking why, if they're recycled, do they ever need to be replenished? They can't be recycled forever. In the same way that enzymes can't be recycled forever. We make new enzymes all the time, replacing old enzymes. Does that answer your question?
Somewhat, thank you.
Somewhat? Okay, what part is missing?
I'm, like, okay, you said earlier about photosynthesis, the animals eat that, and then we get, I guess, we get the energy from the animals because we eat them, right? So we're getting the vitamins and the energy and stuff from the animals that's consumed. Okay, then the vitamins that we get in our bodies, what I'm saying is, you're saying it recycles and everything, but then we have to replenish them because you said now that they don't always recycle because there's an end to it eventually.
Yes, they're not permanent. Enzymes are not permanent. Vitamins are not permanent in the system. You have to continually replenish them because remember the second law of thermodynamics, things break down if you don't keep providing an energy input and making new everything, you eventually run out of materials. The body does.
Okay. All right. Thank you.
Now we're going to talk about the energy molecule, ATP, adenosine triphosphate. Now we've run across this before. Adenine is the A base found in DNA. So adenine, adenosine are components of our genetic code. So that's one fundamental role that this molecule plays. But it has another critical role, and that is in energy metabolism. This picture here shows the structure of an ATP molecule and its components. So adenine is the base shown here at the top. Ribose is a sugar, and the two of them together are called adenosine. If you add a phosphate group to the sugar, it's now adenosine monophosphate, 2 sugars diphosphate, 3 sugars triphosphate. That is ATP, and that is the energy molecule. So ATP links endergonic reactions with exergonic ones. And it's toggling back and forth between the two continuously in all the cells of our body at all times. ATP is being made and ATP is being hydrolyzed continuously. To make ATP, energy goes in. It's an endergonic reaction. And when ATP is hydrolyzed, the terminal phosphate is released, and you get ADP plus PI, and energy is released. So ATP is a high energy molecule. ADP and PI are much lower energy molecules. That's why when this hydrolysis occurs, energy is released, and it's this energy that drives cellular functions. And that brings us to the two different types of metabolic pathways: anabolic and catabolic. Anabolic pathways build up molecules. They link together simple molecules, monomers, to form polymers, complex molecules. The anabolic reactions store energy, so they're endergonic. They require energy to hook together small molecules connected by chemical bonds, which contain energy. And these reactions, as I said, are primarily endergonic. And they require free energy in order to form. Catabolic pathways are the opposite. They tear molecules apart, and when they do, they release energy. They break down complex molecules to form simple ones, and they release energy. And this illustrates and contrasts the two. So an anabolic pathway, remember, builds up, takes small molecules like monomers, consumes energy, and makes a polymer. Catabolic pathways are the opposite. You start with a polymer, and remember this, the energy that it takes is now in the bonds connecting the polymer. And now this high energy polymer is broken down in a catabolic pathway, releasing the monomers plus energy. So if you think about this in terms of overall processes, photosynthesis is anabolic. It takes energy, makes small molecules into large ones. Respiration is catabolic. When we eat food, we eat largely polymers. They're broken down into monomers, and as they are, energy is released, and that's what makes us go every day. And that's illustrated here. Photosynthesis is primarily anabolic in that you start with carbon dioxide and water and energy from the sun that makes glucose and oxygen. The opposite of that is respiration, which is glucose oxidation, and it's catabolic. You take sugar plus oxygen, releases energy, and leaves carbon dioxide and water. So the overall chemistry of photosynthesis and respiration is very simple, but we'll go into the complexities of it when we get to those two topics. All right, pathway regulation. This is essential for a living system to stay in a homeostatic state. In a living systems, most enzymes are not needed most of the time. So either they're not there most of the time, or they're inactive most of the time. And they only go into an active state when they're called upon. So how can enzymes be active only when they're needed? There are various ways to regulate them, usually by inhibitors. And inhibition can be either irreversible, that is, you can't change it back, or reversible, which means you can. So here are types of reversible inhibitors. They can be either competitive or non-competitive. In the top diagram, you see an example of competitive inhibition. Enzyme is this big blue thing. The competitive inhibitor is in a different shade of blue, and the substrate is yellow. Now, the substrate would like to fit, remember the shape is critical. So the shape of the substrate wants to fit into the shape of the enzyme. Well, it turns out that this inhibitor has approximately the same shape so that it can fit into the active site. So if the competitive inhibitor is there, the substrate can't be there. And so it isn't, the enzyme reaction does not occur.
So you're saying that neither one, they both can't go into that, only one can, so only one will be able to fit.
Right, and if the inhibitor is there, there's no reaction because the substrate can't be there. And if the competitive inhibitor concentration is high, then most of the enzyme sites are going to be occupied, and very little chemical or enzymatic reaction will occur. It's only if the competitor, if the inhibitor is either low in concentration or missing entirely, that the enzyme will convert the substrate to a product.
Professor, is there a certain source where you get those inhibitors? in other words, is it something that you intake in your body? Is it something that your body developed by itself? How does this happen?
The inhibitors can be of various types. It's most often it's things that you yourself, your body synthesizes and expressly for the purpose of enzyme regulation. Well, there's another type of inhibition called non-competitive. So here you have the enzyme with its active site and the substrate which fits in it. The non-competitive inhibitor does not bind to the active site, but rather at a different site. But when it does that, it changes the shape of the enzyme so it no longer fits the substrate. So the effect is the same. As long as the inhibitor is bound to the enzyme does not function. My favorite type of regulation is feedback inhibition. I think this is really cool. So you have a pathway starting with the substrate, and you have enzyme 1 converts that substrate to a product. Then enzyme 2 converts that product to another product, and so on, enzyme 3, enzyme 4, enzyme 5. And finally, you get to this red box, which is the end product. Now, if the cell is calling for this red box, the pathway will run. But if the cell no longer needs this red box, this end product, it will accumulate. And when it does, it binds to enzyme number 1. And when it does, it's an inhibitor, and enzyme number 1 can't function, and the pathway stops. You don't get any more end product. So think of this. So for example, if you cut yourself and wound healing has to go on, which is very complex, but supposing this end product is fibrin, a component of scabs. Okay, you've cut yourself, you need a lot of fibrin, so this pathway is going to be running full steam, making as much fibrin as it possibly can. But now as your scab heals, your wound heals, and the scab gets completely formed, the need for fibrin is reduced, and finally you don't need any more at all. And so this end product, fibrin, can bind to the first step of the pathway and shut the pathway down. And this pathway remains off then until the next time you cut yourself or you need to heal some other kind of wound. So I mentioned that just as a perhaps way for you to visualize how feedback inhibition works. When your body is calling for a product, the pathway runs. When it doesn't need the product anymore, pathway shuts off. And the reason it shuts off is the end product inhibits the first step.
I have a friend that's dealing with wounds not healing properly. So can it be that that process can end before it should, and cause that wound not to get scabs or what have you? Or to
Could be,
slow?
more likely he's deficient in one of the enzymes. Yeah, it's hard to know. It could be an enzyme deficiency. It could be that that enzyme number 1, in his case, is particularly sensitive to end product inhibition, shuts down prematurely. You'd have to have an analysis in order to know.
And you have to have
Thanks.
all 5 of those to get that scab he's referring to, right?
Say that again.
You have to, and like what he was saying, to get that crust over your wound, you have to have all 5 of those enzymes to get that end product, to get
Yeah,
that
you have to have
crust.
a full pathway. It may not be these 5 steps. It could be any number of steps, but you have to have some way of converting substrate to product. And when this product is needed, the pathway is going to run. When the product isn't needed, it accumulates and shuts down its own pathway.
I know you just said that you could have any number of steps, but do you really mean like could it make like 15 different enzymes before it gets to an end product, or is there a limitation to it, or is it just going to whatever the final product needs to be, so it could happen like thousands of times?
Good question.
Yeah, the number of enzymes needed in a pathway is highly variable. It could be from 1 to many. I don't know that there is an upper limit to the number of enzymes in a pathway. Some pathways are very long.
Okay, I see.
All right, let's go on. And now we have to talk about redox reactions. And where this is leading us to really the heart of the problem. Energy is harvested through electrons. And these electrons move from one molecule to another. And as they do, they carry energy. And the basis for the transfer of electrons is called redox reactions. So some substances attract electrons more strongly than others. A substance that loses an electron is said to be oxidized. If it gains an electron, it's said to be reduced. And these reactions, oxidation and reduction, always occur in pairs. And as I said, they're known as redox. So let's visualize this. It's really important that you understand how it works. So we have here substance A, substance B. Substance A is reduced. That is, it has an extra electron. So it's reduced. If it transfers that electron to another molecule, in this case B, substance A is now oxidized and B is reduced. Now there's this little mnemonic called oil rig, which you might find useful in helping you to remember this. Oil stands for oxidation is loss of an electron. Rig, reduction, is gain of an electron. That may or may not be helpful. Some people use it. So let's make this into a biological situation. We have an organic molecule here that is reduced. It's got 2 extra electrons. And these can be transferred to what's called a coenzyme, which is an electron carrier. And when that happens, the organic molecule is now oxidized and the carrier is reduced. It has 2 extra electrons. And so we can think of this in terms of handoffs from one to another, which becomes a chain. So we have substance A, which is reduced with 2 electrons, 2 hydrogen atoms. It passes them off to NAD, so it becomes NADH plus H, which is energy. NAD passes them on to substance B, which is goes from oxidized to reduced. And this carrier, NAD, is found in animals in glucose oxidation. If it's in plants, if it's in photosynthesis, it's NADP. It's a different molecule, but does the same function. So this just shows you the structure of NADP. It's a very complex molecule. It's a huge enzyme. It contains 45 polypeptide chains. A little hard to imagine that, but so be it. So it can be either oxidized NAD plus in this form, or reduced where it takes on an extra pair of hydrogen atoms or protons. And this all comes down to what's called the electron transport chain. This is where we get our energy. So electron transport chain is a series of proteins that are in the membrane. And I'll talk about which membrane in a minute. But they transverse across the membrane from inside to out, and there's a whole series of them. So electrons are transferred then from NADH, which becomes NAD, but they're transferred to a carrier known as a flavin, to another carrier within the membrane, which transfers to another carrier within the membrane, and another, a series of cytochromes. And finally, as these electrons are transferred, they're giving up energy. And that energy is in the form of protons. So the electrons are transferred, they're giving off protons, which are accumulating. So in this case, we have 6 hydrogen atoms, and these then at the very end, the final electron acceptor is oxygen. So you take 1/2 of an oxygen molecule, it accepts electrons and becomes water. So if you start out with food, you end up producing water. And where does the energy come from? It's an ATP synthase complex called chemiosmosis. So the protons that are generated by this electron transfer chain are passed through the membrane, back the other way, through the membrane, through this channel. And when they do, this enzyme converts ADP to ATP. And this is known as chemiosmosis. So let me just go back to this, where I was trying to make the point that electrons are transferred from one molecule to a carrier to another molecule. Instead of one carrier here, we have a whole series of carriers in the electron transport chain. So each of these carriers shown here is one of these steps. And in the end, we start out with a reduced compound. We end up with ATP. And this illustrates chemiosmosis in more detail. Again, we have a membrane. We have these transmembrane channels. We have the electron carriers transferring electrons from one to the next, to the next, to the next. And every time an electron is transferred, a proton is produced. So the protons keep accumulating in the intermembrane space. They originate in the matrix and end up in the intermembrane space. And now there's a high concentration of protons outside the membrane, and they get transferred through this channel, this ATP synthase called chemiosmosis, and they convert ADP plus PI to ATP. So this happens in either the chloroplast or the mitochondrion, and this is how we get energy, biological energy. Well, where does it happen in a cell? Well, here's a cell, here are the various organelles, and here is the mitochondrion. So you take a closer look at the mitochondrion. There is an inner membrane, an outer membrane, a matrix, and the chemiosmosis occurs in this inner mitochondrial membrane. So the protons are transferred from, the electrons are transferred from carrier to carrier within the membrane. Protons are transferred from the matrix outside, and then when they go back across the membrane by chemiosmosis, they generate ATP. Now, why is all this complexity necessary? Why not just take the electrons from glucose and convert them to water and carbon dioxide and release energy? Well, because it would cause, there'd be a huge release of energy that the cells could not capture. So there has to be a way to slow down the electrons so that energy can be captured. And that's done in an electron transport chain. So you see that illustrated here. So instead of just going bang from glucose to water and carbon dioxide, it goes stepwise, much more slowly. So you start with glucose, plus oxygen, and you release carbon dioxide and electrons. And these electrons, as I illustrated, these electrons are transported through these carriers, and as they go, they release protons. And so you can think of them as going down steps, because at every transfer from carrier to carrier, they lose energy. The electrons are losing energy, but it's being maintained in protons. So energy input splits glucose into carbon dioxide, oxygen, and electrons. Electrons lose energy as they move down the electron transport chain, and energy is released. The energy released by electrons is harnessed into cellular energy. In other words, in ATP. Okay, I'm out of time, so let me just briefly mention one thing. There are three ways to produce ATP. And don't worry about this right now, because we're going to talk about each of these processes in detail. But there's substrate level phosphorylation, there's oxidative phosphorylation, and there is photophosphorylation. Now they're illustrated here. So substrate level phosphorylation converts ADP to ATP, and it's a 1-to-1 ratio. You get 1 ATP from every ADP. Oxidative phosphorylation is much more lucrative. It produces many ATPs from a single molecule of glucose. You can get 38 ATP molecules. So you get a lot of energy from oxidative phosphorylation, and this occurs in the mitochondrion. Photophosphorylation occurs only in plants. The energy for ATP production comes from light, but again, you have to have an electron transport chain. And in both cases, chemiosmosis occurs to convert the energy in the protons into ATP. All right, I am slightly over time. And that was a lot. And we're going to do reviews of this information before the test, because it's complicated and it's a lot to assimilate. And this set is only about enzymes. Next, we're going to talk about photosynthesis, and then we're going to talk about respiration. Again, complex processes that we'll talk about in detail.
So we won't have the test Monday? The quiz?
No, Monday we'll talk about photosynthesis. Wednesday we'll have a quiz. The quiz will cover molecular genetics, this lecture, and photosynthesis. So that'll be next Wednesday, November 17th.
For the quiz, okay.
And then we're going to have a discussion period where we can discuss everything that we've had so far. And then, well, we'll see how it goes. I don't know how fast we're going to be able to go through this, but we're off the week of the 22nd, I believe, of November. And then we come back for, I think, just 3 periods in December. And one of those will be committed to test 3. And then the final exam will be the following week. And I tentatively said Wednesday, December 15th for the final exam. So unless I hear complaints about that, that's when it will be.
Okay, well, have a good weekend.
Okay, this is hump day, so it's
Have a good weekend.
all right.
Bye-bye, guys.
Yeah, we'll see you next week. Have a good day.
Bye, Yolanda.
Bye. Have
Bye,
a good
everyone.
day.
Stay safe, guys.
Thank you,
Bye.
Professor.
Driving.
Bye.
Bye.